It's truly a pleasure to be able to present our work using cytoph to characterize innate immune dysregulation, particularly NK cells, to this audience. This is work that was published late last year. So, juvenile dermatomyositis is a debilitating, multi-systemic pediatric autoimmune disease manifesting with characteristic rash and proximal muscle weakness. It's both adaptive, as Anne just talked about, both adaptive and innate immune responses have been associated with JDM pathogenesis, but the dysregulation of immune cell signaling in JDM has not been systematically studied. Prior to the use of steroids, JDM had a mortality rate of 30 to 40%. Current treatments, even with current treatments, the disease inflicts significant morbidity on children, with a substantial fraction of JDM patients experience a protracted refractory disease course. And we believe that a better understanding of the etiology of JDM may lead to new targeted therapies. I originally was going to talk more about pathogenesis, but I saw that Anne was going to do that, and she gave a very nice comprehensive review, so I'm just going to jump right down to NK cells. So NK cells are innate, usually different. NK cells are on. Sorry. Yeah. Okay. All right. Let's get back on. Is this not working still? Okay. We'll just use the mouse then.
Very difficult to see on the right.
Forget it then. Fine. So, NK cells are innate lymphocytes with germline encoded receptors that play a critical role in antiviral host defense and tumor surveillance. And this diagram there on the left side in the innate component. They're large granule bone marrow derived innate lymphocytes that are identified as CD56 positive, CD3 negative. They're distinguished from T and B cells by the lack of rearranged antigen receptors, and they comprise 5 to 15% of our circulating PBMCs. NK cell responses are regulated through interactions of germline encoded receptors, both inhibitory and activating. So the blue receptor on the bottom is representing an inhibitory receptor, and that binds to MHC class 1. So essentially, NK cells are going through and surveying self, self, self. And so cells that downregulate class 1 are more vulnerable to NK cell killing. There's also activating receptors, shown by the green receptor in the top, and they bind to stress-induced ligands and virally encoded ligands. And then the NK cell chooses to respond by integrating signaling through all these different receptors. And that signaling level, thank you. Let's see if this one works. Oh, beautiful. So that signaling threshold can be modulated by the cytokine milieu. So when an NK cell gets a strong signal through its activating receptor, particularly in the absence of an inhibitory signal, it responds and it mediates these effects by releasing cytolytic granules that contain granzyme and perforin to be able to lyse the target cell, by releasing immunomodulatory cytokines or chemokines and proliferating. So just a little bit more about what's in this black box after the activating receptor signaling. And this is a schematic I took from another paper. If you just look at the left first, so when an NK cell encounters a target cell that it finds, there's a signal on that's abnormal, it forms an immune synapse, and then there's some signaling that goes on, and then the cytolytic granules move to the microtubule organizing center, the MTOC. The MTOC moves to the synapse, and then these granules confuse and then lyse the cell. If you zoom in on the signaling part, again, you have activating receptors, some proximal tyrosine kinases, but very central in NK cell responses is a protein called phospholipase gamma G2, or PLC gamma 2, and that'll be important for the rest of this talk. So NK cells are able to modulate innate and adaptive immune responses, and there's accumulating evidence that human NK cells play an immunoregulatory role, and that NK cell dysfunction may contribute to the onset of human autoimmunity. So in regard to specifically what is known about NK cells and JDM, Anne touched on some of these. There is decreased, several reports have demonstrated evidence of decreased NK cell percentages in the blood of JDM patients compared to controls, including both the study from Dr. Pockman and the study from Anne. There's some preliminary evidence that there's increased NK cells in the muscle biopsy from JDM patients. I think the strongest data, which is still pretty small groups, but is that there's decreased NK cell cytotoxicity in JDM patients in 2 small studies. So there was 5 patients on prednisone in this early study, and there was 2 treatment-naive patients also in an early study. So my lab hypothesizes that innate immune signaling dysregulation may contribute to the onset of dermato. And to delineate signaling abnormalities in PBMCs from patients and controls, we utilized mass cytometry, or CyTOF, coupled with antiphosphoprotein antibodies. So I assume that most of you are familiar with flow cytometry. So mass cytometry is similar in some ways, but the major difference is that rather than using antibodies conjugated fluorochromes, it uses antibodies conjugated to heavy metals. And then the labeled cells are nebulized in single-cell droplets and ionized and analyzed in mass spectrometer, but you get data out that's very similar to flow. The real advantage is, if you've done flow, is that there's a lot of spectral overlap, so it limits the size of panels you can have. And there are getting to be flow machines that can use larger panels, but in reality, most things are done with 10 to 12 colors. In contrast, there is no spectral overlap with these heavy metals, and there's really no compensation. So you can use panels of 38 to 40 markers. So this is particularly valuable in small samples like blood samples from pediatric patients. So we investigated signaling abnormalities in a non-biased way, just broadly. Even though I'm interested in innate immune dysregulation, we just looked at signaling across 23 different immune subsets. In 17 treatment-naive JDM patients, 11 of these 17 patients after achieving clinically inactive disease, and 17 healthy controls. And this work was done in collaboration with Dr. Pockman. And we validated and used a 37 antibody panel for CyTOF, including 20 antibodies to subset all the immune cells, and that allowed us to distinguish 23 immune cell subsets. And then we had 15 phosphoantibodies to evaluate signaling. And this allowed us to simultaneously gain insight into multiple signaling pathways. Samples were stimulated with IL-2, IL-12, interferon alpha, and LPS, in addition to IgM, CD3, and CD16 cross-linking antibodies for 0, 3, or 15 minutes, and then subjected to mass cytometry. In addition to delineating signaling differences, we were also able to garner information about the relative frequencies of different immune cell subsets, and I'm going to show that first. So there's 23 immune cell subsets shown here. I've broken them into the larger subsets and smaller so we can have different scales. And you can see some of the same things that Anne had talked about. There is increased naive B cells in treatment-naive patients. So the treatment-naive patients are on the left, they're the open circles, compared to controls. There's increased naive CD4 T cells, and there's decreased percentages of NK cells in the periphery. So we were interested in what happened in the 11 pairs we had pre-treatment and in clinically inactive disease. And so we looked at those, and there was decrease in the B cells. There's also non-statistically significant, but decrease in the naive CD4 T cells, and an increase in the NK cells, which was also not statistically significant after correcting for these 23 comparisons. But 9 out of the 11 paired NK cells went up, and the level they went up to is indistinguishable from the controls. There was other things on our panel that I'm not going to have time to talk about, but I'm just going to touch on. We looked at markers of activation and proliferation. I'm just showing this for NK cells, that even though there were lower percentages of NK cells in the periphery, they were more activated and they were undergoing substantially more proliferation. So back to the main purpose of this study, we assessed 13 phosphoantibodies in 23 immune cell subsets at 0, 3, and 15 minutes following stimulation. This results in a very large data set. So there were 299 features, and I'm going to use the word feature several times. So it's a specific phosphoprotein, phosphorylation of a specific signaling molecule in an immune cell subset at each time point. And so you can't just look and say what's significant. You have to do some math. And so we used a program called Citrus with last cell reduction. And so I'm just going to summarize this briefly by saying Citrus is a technique that combines unsupervised hierarchical clustering with a regularized supervised learning algorithm. And LASSO is a program that enhances generalization by reducing overfitting and removing redundant irrelevant terms. And this approach identified 12 features, again, phosphoproteins in a specific immune subset that distinguish the treatment-naive patients from the controls. And surprisingly, PLC gamma 2 phosphorylation differences comprise 10 of these 12 stratifying differences that distinguish the two groups. And as I've implied earlier, PLC gamma 2 is a particularly critical enzyme in NK cells and B cell signaling. So T cells primarily use PLC gamma 1, but they express PLC gamma 2. This is a busy slide, but let me just walk you through it. This is the heat map of the arc-sign transformed median signaling molecule intensity of the 12 stratifying signaling molecules at specific time points. And so the signaling molecules are designated in the rows, and you can see what they are on the left. And the columns represent the 17 treatment-naive patients and 17 controls. And you can see at the top that these 12 stratifying features were able to completely separate the patients from the controls. I think this is easier to see on a partial least squares discriminative analysis. So if you look at the scores plot on the left, which shows the relationship between the samples, you can see that the patients in red are completely separated from the controls in black by these 12 stratifying features. If you look at the loads plot, which is on the right, it shows the relationship between the different variables. And you can see that NK cells made up 5 of the stratifying features. PLC gamma 2 made up 10. But you can see that in NK cells, at every time point, they had decreased phosphorylation of PLC gamma 2. Surprisingly, some of the stratifying differences were hyperstimulation of T cells. And I don't have time to really talk about that today. So we wanted to confirm that this was real. The student working on this was a math major, was an undergrad and a BME graduate student, and I made her analyze this several different ways. And so one of the ways we did this to confirm was to use, the significance was use two-tailed Welsh t-test with stringent Bonferroni correction, so we corrected for the 900 comparisons. And this statistical test identified the 3 most significant features, again, phosphoprotein in a specific immune cell, as being NK cell PLC gamma 2 at 0, 3, and 15 minutes. And another one is MapCap K2, which is a downstream from PLC gamma 2. So this really highlights the importance of dysregulated PLC gamma 2 signaling in NK cells in this disease. And therefore, the rest of this talk, I'm really going to focus just on the NK cell work to see if this makes a functional difference. So this is a schematic, again, of the signaling pathway where you get NK cell activation receptor and ITAM, and then some proximal tyrosine kinases and PLC gamma 2. And so when we went back and gated manually, just like you would on flow, so just gated manually on the whole NK cell population, we saw the same difference. So the patients are in the solid line, and the dotted line is the controls, and this is the median intensity of PLC gamma 2, normalized to a run control. And they were statistically significant at each point. In contrast, if we look at proximal tyrosine kinases, including SYK and ITK, there were no differences. And when we look downstream at MapCap K2, it mirrored what we saw in PLC gamma 2. And what happens when we looked at the same patients when they reached clinically inactive disease, they were intermediate between the controls and the patient and were substantially normalized. And this was also seen when we looked downstream at MapCap K2. So is this decreased phosphorylation just because there's less PLC gamma 2 there? And we did look at both at the protein level and the transcript level, and we did not see any differences. So I want to just focus again on the schematic we talked about before. So PLC gamma 2 occupies this spot. Its job is to cleave, when it's phosphorylated, it cleaves PIP2 to IP3 and DAG. IP3 goes and binds to the endoplasmic reticulum and releases calcium, which causes calcium flux, which initiates this whole process where they initiate the cytotoxic granules move to the mTOR moves to the synex. DAG activates PKC and also is involved in localization to the membrane. IP3 has a very short half-life, so we decided to measure calcium flux to see if this phosphorylation defect had any functional relevance. And to do that, we enriched NK cells, labeled them with a dye, and then stimulated them with antibodies to NK cell receptors, so 2B4 and NKG2D. And you see on the left side, here's a calcium flux in a control, and here's calcium flux in 2 of our treatment-naive JDM patients. So this strongly implicates dysregulated NK cell PLC gamma 2 phosphorylation and resulting in decreased NK cell function responses, and provides a mechanistic explanation for why NK cell cytotoxicity may be depressed in treatment-naive dermato patients. So I showed you some data early from the 80s where several different groups had shown that, but I have not directly shown this in this study. So I would like to just summarize this part of what I wanted to talk from that study. So treatment-naive JDM patients have decreased percentage of NK cells, increased frequencies of B cells and T cells in their PBMCs, and these frequencies substantially normalized in paired patient samples with cessation of active disease. CyTOF coupled with a panel of phosphoantibodies revealed dysregulated phosphorylation of PLC gamma 2 in treatment-naive JDM patients, NK cells. Strikingly, 10 of the 12 signaling differences were identified as stratifying were in JDM patients, and controls involved PLC gamma 2. And this hyperphosphorylation of NK cell PLC gamma 2 was the primary signaling difference distinguishing these 2 groups. And no differences were detected in upstream kinases or the total PLC gamma 2 protein levels in NK cells. This hyperphosphorylation in NK cell PLC gamma 2 and downstream MAPK2 was substantially normalized in treatment-naive JDM patients with cessation of active disease, supporting a potential role for dysregulated NK cell PLC gamma 2 signaling in juvenile dermatomyositis. And the suppressed PLC gamma 2 phosphorylation in treatment-naive patients resulted in decreased calcium flux, suggesting that the signaling defect had functional consequences. And so we believe that this novel identification dysregulated PLC gamma 2 phosphorylation and decreased calcium flux in NK cells from treatment-naive JDM patients provides potential mechanistic insight into the etiology of JDM. And I wanted to thank the parents and the patients and their families who participated. This work was primarily done by Alison Throm, who did her biomedical engineering PhD in my lab. She defended last August. Josh Allinger is an MD PhD student in my lab in immunology, and he helped with some of the work, particularly with the calcium flux, and he's defending his thesis in 2 months. As I mentioned, this was done in conjunction with Lauren, in collaboration with Lauren Pachman. The funding was from the NIH, the Strategic Pharma Academic Research Consortium grant, and CureJM funding to Lauren. So I'm really excited about this data, and my hypothesis, and this kind of relates to what Brian asked, is I don't doubt that, given the data that Anne highlighted, that this is an autoimmune disease. By that I mean it's T and B cell driven. But my hypothesis is that dysregulated PLC gamma 2 signaling leads to decreased NK cell function responses, resulting in the potential loss of potential NK cell suppression of autoimmune T cells early in the development of JDM. And in the few remaining minutes, I'm just going to talk about some of the exciting things I think that would be future directions that we want to do to nail this down. And so we want to define the impact of PLC gamma 2 hyperphosphorylation and NK cell function responses in treatment-naive and clinically quiescent JDM patients, as well as adult DM patients in comparison to healthy controls. Because I strongly suspect that we're going to see similar things in adult DM, but we haven't tested it yet. And so that involves defining the impact of this hyperphosphorylation on NK cell functional responses by using a dedicated cytoph profiling of NK cells. So not the panel we use for signaling, but a panel that's much more focused on NK cells, treatment-naive JDM and dermato, and to actually test the functional responses. So I looked at calcium flux, but look at degranulation, cytotoxicity, and cytokine production. And the real limitation for this is just samples, is enough PBMCs to do these studies from treatment-naive patients. I've mentioned briefly that there was evidence of hyperphosphorylation of PLC gamma 2 on T cells, and we're interested in investigating that by doing functional T cells studies and single cell RNA-seq with TCR-seq to investigate if PLC gamma 2 hyperphosphorylation increases the activation of a subset of T cells in dermato. And lastly, we're interested in understanding how transcriptional alterations in cytokine exposure may influence NK cell PLC gamma 2 hyperphosphorylation observed in these treatment-naive patients. So first, we plan on interrogating transcriptional changes in the regulatory components of PLC gamma 2 using RNA-seq of sorted NK cells, B cells, and T cells from treatment-naive patients and controls. And then we're interested in investigating the impact of interferon alpha and other cytokines that, as Ann talked about, are in the milieu of new onset dermato to see if we expose normal NK cells from normal donors, if they also have decreased phosphorylation of PLC gamma 2. And the flip side, we're interested to know if we can ameliorate this problem, if we can with IL-15 or IL-2. And this is based on some data from Josh's work. So Josh, the other grad student, is really interested in looking at patients with unusually severe and recurrent herpes virus infections. And in 2 families, we've identified haploinsufficiency, so mutations in 1 copy of their PLC gamma 2 protein. And this leads to suppressed but not absent calcium flux. So here's one of the patients, and here's the control. These are also very small samples, and so we were expanding them with an IL-15 feeder cell line and incubated them with IL-2 for 7 days. And this isn't published yet, but to our surprise, it really ameliorated this difference. And IL-15 is used clinically in some cancers, and so this is a potential that I'm really interested in examining. And the last thing I'm going to just mention is that, NK cells percentages are low, even though the cells are proliferating later more. That could be because they're apoptosing or they're moving someplace else. And so we're hypothesizing that dysregulated NK cells move from the blood to the afflicted muscle tissue early in development of JDM. And in collaboration with Dr. Pachman, I plan to evaluate muscle biopsies from 7 of the treatment-naive patients that were in our original cytoph study using imaging mass cytometry, and that should be IMC, not IML. So IMC is a new cutting-edge technology that combines high-dimensional aspects of cytoph with the spatial resolution of more traditional imaging techniques, allowing the visualization of 37 to 40 markers in the spatial context of the tissue microenvironment. So WashU just acquired one of these machines in November, and they're getting it all lined out, and I think it's going to allow really interesting studies. So this is a schematic I took from a paper, the people that first pioneered this. And so tissue biopsies are labeled with metal-conjugated antibodies. Then regions of interest are designated, and they're ablated with a laser going 1 micron by 1 micron at a time over a 1 millimeter by 1 millimeter region of interest. And then they're put through a mass cytometer, essentially through the cytoph, and you can keep the data on the localization of and still do the deep profiling, as I've shown you that we've done with cytoph. I mean, you can't do signaling because these cells are fixed, right? But I believe you will be able to do some really interesting things. So we'll be able to delineate the composition of immune cell infiltrating muscle biopsies from children with juvenile dermatomyositis at a level of detail that's not been previously possible. We'll be able to characterize the phenotypical differences between the infiltrating immune cells and the immune cells in the blood. And you'll be able to assess which infiltrating immune cells are in direct contact. So we're excited about this project. But thank you for your attention, and I'd be happy to answer any questions.
Tony, I suppose you addressed it when you were talking about the IL-2, IL-15 experiments, but how do you suppose it is that this phosphorylation defect is being addressed and treated with our current treatments?
I'm not sure. I actually don't believe that NK cells are the drivers here. I want to say that right front. So I think later in disease, it is corrected or substantially corrected, but I don't think it's the driver anymore. Prednisone knocks it. Yes, so, yeah, they were substantially normalized. I'm not sure of that answer, Brian. I don't, and in fact, we really don't understand why it's low to begin with, and I think that might let some insight into that. If it changes the transcription of something, or we just knock down interferon alpha, and now whatever's driving changes in regulation now goes away, but I don't know. Hey, Ann.
Hi, I have a couple questions. One, I thought you were addressing is this specific to myositis, or do we see this in other disorders that have inflammation or something similar? My second question was more to speculate on is, you know, when we talk about percents of cells and we talk about all these things and even these abnormalities, we're often looking, you know, we're saying the percent is up or the percent is down, but what does that mean? It could be the absolute
Yes or
number
no.
is just fine and something else is down,
Yeah.
and that's why the percent looks off. And have you looked at that?
So that question, that's an important question, and we have changed the way we're harvesting cells now to do essentially a CBC in lab at the same time, because you never know when you just have percentages. This is always a problem, and if you don't have, even if we have a CBC on the patient samples, we don't have a CBC, a clinical CBC, we don't have a CBC on the controls. But this is a problem, and we're addressing that by the way we're harvesting cells. And the first question was what again?
Do you
if
see
it's
these
specific.
phosphorylation changes in
Yeah.
any
So,
disease?
I don't know because I've not, actually viewers wanted it in lupus, JDM, and polycythemia JA, and we just haven't done that. This is, I mean, new technology and it's very expensive. We do see it in patients that have recurrent severe herpes virus infections. Not this suppression like this that gets better, but that at least these two families had mutations. I think it reflects the fact that it's very important. NK cells don't have very high levels of PLC gamma 2, and it's very important that when an NK cell gets a stimulus to kill another cell, it's real. It's not, there has to be a strong signal, but I don't know how widely expressed this is. I suspect that it's not just in myositis.
Thanks.
Tony, have you had a chance to think about other regulators of calcium flux in the neighborhood?
So there's always calcium flux. It's not just through PLC gamma 2. That's very clear. And I have not, because what we saw as distinguishing or stratifying these two populations was in there. But there's certainly going to be other molecules that calcium flux is regulation is very complicated. I agree with that.
It's very good.
Yeah.
Really elegant discovery, Dr. French, thank you. Question about, do you think that you might find that PLC gamma secreting defect more frequently in the polycyclic chronic continuous group of JDM patients as opposed to the monocytic ones? For that one sentence you said about it allowing, not being there to suppress the autoimmunity.
Yeah. So this is completely, just guessing. I don't have data to show this, but I don't think it's the NK cells killing the muscle tissue. I could be wrong. I think it's more that they have allowed autoimmune T cells to escape regulation. And so I don't think it's, we know that actually I don't. I think once it gets going, it's not the NK cells anymore, is what I think. But that's conjecture. I don't have data for that. Yeah.
We know that NK cells, and you especially know, that are really important activation syndrome,
Yeah.
which can occur in JDM, but it does really rarely occur in other diseases. If NK cells are really involved, why do you think that?
So that's a good question, Amir. Why don't they get it at the beginning? I would guess it's partly due to the cytokine milieu that they're in here, that it must not lead to that, but that's a good question. Thank you for your time.